This method can help answer key questions in the HIV entry field, such as drug discovery and identification of factors required for HIV fusion.
The main advantage of this technique is that it can be scaled up for high-throughput screening.
This technique can be used to screen large libraries of compounds to identify drugs that target early stages of the HIV-1 life cycle.
This method can also be used to study HIV fusion inhibitors in animal models, such as humanized mice.
Researchers should follow biosafety precautions with infectious HIV-1.
This is a recombinant full-length viral clone that does not replicate in multiple rounds, but must be approved by your biosafety office.
To begin this procedure, thaw one 500 microliter vial of Jurkat RG reporter cells by placing it into a 37 degrees Celsius water bath. Pipette the cells from the vial into 10 milliliters of RPMI complete medium, and then centrifuge the mixture at 800 times gravity for 5 minutes at 23 degrees Celsius. Resuspend the pellet in 20 milliliters of RPMI complete medium and transfer the cell suspension to a T75 flask. Incubate the flask at 37 degrees Celsius overnight. On the following day, add 0.5 micrograms per milliliter of purimycin to the cells in the flask. Culture the cells maintaining a density of 200,000 to 800,000 cells per milliliter. On the day prior to setting up the transfer assay, split the cells down to 200,000 to 400,000.